So good afternoon to everybody. So I will try to perform this, presentation more informally that I was scheduling in my mind because I couldn't have the opportunity to attend the two previous sessions that has been done because I was too busy to do in bureaucracy and papers stuff. So my name is Lisa Vaccari. Just since everybody introduced a little bit of itself, I'm very old, so I will do a short introduction. I'm an analytical chemist in the origin, but basically for all my life I'd worked in the Sincrotrone facility. In the first period, I was working mostly on micro and nano fabrication approaches, and then from 2006 I'm working at the beamline that is called SISSI. So beamline means a lab in the synchrotron, essentially, that exploit synchrotron radiation, and the name is SISSI, Synchrotron Infrared Source for Spectroscopy and Imaging, and especially for chemical and life science application. So in today, I want to give you a flavor of the things that can be done with at the beamline, and specifically with the topic of vibrational cytology and cytology and histology. That is something that fits with the other techniques that has been considered in the PRP CERIC project. So this I can skip, just to introduce you something. So I'm totally unable to speak. Can I stand up? Okay, it's impossible for me. I'm too energetic and I need to move. So SISSI, as I told you, is the acronym for Synchrotron Infrared Source for Spectroscopy and Imaging. So as you can imagine, we use infrared synchrotron radiation. Okay, so the light that you see in that part is the visible light that comes out from the beamline, and how it is propagated on some mirrors that we like to play, in the lab to see how things are moving and going. Yes. The point is the following: the beamline is divided into branches. One branch is called SISSI-MAT, the material and chemicals, the material science branch line that is managed by the CNR. Okay, that is not the matter of this presentation. And the other is the SISSI-Bio, that is the branch line that is managed by Elettra. This is mostly dedicated, as I told you, to chemical and life sciences. So essentially, I think that the very important thing to highlight is what we do at SISSI. So how it is used infrared light. So the energy of infrared light is basically comparable with the energy of the vibration in the molecules. Okay. So what you probe is how things vibrate. And in order to have a vibration, you have to have atoms, masses, okay, and linked together with chemical bonds. So we probe the chemistry essentially of things by knowing through the energy of the vibration which type of molecular groups are vibrating, okay, and how many of them we have in a sample. So essentially we do this. The very special thing that we can do in the lab, and it will further implemented with the PRP CERIC project, is the fact that we can assess information, okay, on the materials at different length scales. So we can have conventional spectroscopy, means having average information on a sample without any lateral resolution. We can perform infrared microscopy, means that we can visualize our sample and having chemical information with a lateral resolution of a few microns, okay? Or we can go further, that is what is called here infrared nanoscopy, that is this guy here. So basically we can push the lateral resolution up to having information on the chemistry of the sample and with the nanometric lateral resolution. So essentially what passes from this situation to this situation is that you have two different types of microscopies. This is what is called the far field microscopy. That is also what all of you are quite, used, okay? That is the one of the classical optical microscope. So essentially lateral resolution is comparable with the wavelength that you are using for probing your sample. So infrared is in the micrometer wavelength, and then micrometer is lateral resolution they can achieve. This is a type of microscopy that is more, that is called a near field. So the rules are not the same of the far field microscopy, but is the lateral resolution is given by other physical phenomena. And it has a very interesting thing that while this lateral resolution depends on the wavelength, so depending on the energy of infrared I'm thinking, I have different lateral resolution. This guy here is fully independent for the wavelength. So you have the same lateral resolution for in the entire infrared range. So we work with the infrared synchrotron radiation, but we also work with the conventional sources. And these conventional sources can be glow bars, okay, or lasers, okay. So since maybe it's completely out of your scheme of knowledge, if you want to stop me and ask things during my presentation, please do it. So I feel very informal in this stage. It's just knowing each other, okay. So essentially, which type of things or okay, maybe one thing that you don't know, I think that there was the Barbara Rossi seminar, but I want just to recall you that a beamline in a synchrotron is not a private lab, you mean, like university lab. So we basically are open to what we call users. So people all around the world, they submitted a sort of idea of experiment that is called proposal. This idea of experiment is reviewed by an international peer review panel, and the ones that are better scored, they have access to the facility. So essentially 70% of our time is dedicated to the user and 30% of our time to maintenance and in-house research. So essentially we are a research infrastructure that provides time to user. Our entry points are okay, Elettra, obviously, because we are in Elettra, but we have also an entry point through CERIC, okay, and also more recently is the entry point through an NFFA, that is also subject of another PRP project area, is not leading in this case, but participating. Let me know.
Yes. Wanted to tell you that they cannot see you if you go over there.
I see, okay, but
just to like
doesn't matter.
see you. Doesn't matter.
I'm not so nice. I'm bold. Come on. I'm not a big deal, exactly. So that's a little bit of the story. So among our users, okay, this is more or less the distribution of the topics of the experiments that they propose to us. So most of them, they belong to the life science domain, and then I will talk more about this. We have a community on the environmental science that for a certain extent is also a type of life science nowadays. So there is a very crosstalk between our life and the life of the environment. And we have a community for the chemical science, and in particular, the cultural heritage community that is becoming more and more important in these last years. So, considering the type of experiments that we do with the life science domain, we work with cells and tissues, and we work also with individual molecules. And I will try to provide to you some examples in order to try to understand which is the crosstalk among the possibilities on the possible other approaches in the PRP CERIC. So I started with this one because it's something that can be maybe more understandable for you. So, okay, as I told you, infrared probes vibrations, and with vibration, I know which molecules are vibrating, okay? Because I know that this spectrum, that it's very boring, but it provides to you a lot of info. There are some absorption bands, and these bands are related to the molecular constituents that are vibrating. This is the spectrum of a protein. The point is, most of the time when people know infrared just very roughly, I would say just from scholastical point of view, okay, I know that they think that you can just use it for recognize something. But this is just a small fraction of the use of infrared analysis, okay? You can do other things. The other things that you can do, for example, is to retrieve information from the spectrum about the, I would say, for a protein, for example, about the secondary structure domains that characterize a protein. It's more similar if you are a little bit more confident with UV information that you can obtain from UV, okay? And why can you do this? Because I told you, when you have a vibration, things are vibrating. So the position of this vibration depends on the atoms involved and the strength of the bond. but also it depends from the dipole momentum of the system. So how the charges are distributed. When you have a secondary element of a protein, like alpha helix or beta sheets, if you think about what is giving these different domains is the network of hydrogen bonding. But hydrogen bonding is a sort of electrostatic bond that changes, the separation of charges. So accordingly, with a different pattern, you can have different position of these bands. So you can obtain information if you have a alpha helix, beta sheet, random, and so on, and how they change accordingly, to some stimulus that you can apply. So what I want to show you is also the following. So you have also a couple of very big limits when you use infrared. The sensitivity, because most of the time these techniques is limited to millimolar sensitivity, that is not really used for most of the time for physiological concentrations that are of interest, like more on the nanomolar regime or micro, nano, even femtomolar regime. And the second one is the water barrier. Unfortunately, water is a very, very strong infrared absorber, okay? And one of these is band, is the banding of water, that is this band here, overlaps with the band of protein that is of my interest for knowing about the structure. So I will make the story shorter, but this is something that I already show also at your workshop, but there are strategies in order to enhance the information that you can achieve. And these strategies go through the use of plasmonics. So this merges my basically nanofab, past with my biospectroscopy present. So you can build some nanostructures that most of the time are nanoantennas. These are like gold nanoantennas that are made in a length and thickness in such a way that they act as a concentrator of the electric field. So the signal that you can obtain, infrared signal, is proportional to the incoming electric field. So if you can squeeze your electric field and increase the incoming energy, also your signal will be higher in the same way, okay? So these are like concentrators of energy, okay? So if you are able to put your protein exactly on the points where the electric field, okay, is enhanced, you can obtain a signal enhancement of 6 to 9 order of magnitude. What does it mean? This means that you go from the molar, micromolar to nano, micro, from millimolar to micro, nano, even femtomolar sensitivity. So you can overcome the sensitivity limit if you use such a kind of structured devices to from your body. It's not very different. You are maybe more familiar with the Vaccari system. Okay, it's exactly the same principle of the Vaccari. These are plasmons, but the Vaccari using light, visible light, okay? In this case, you use infrared light. So you have two types of information, essentially. If you have a protein and you have a binder, if you look at the shift, okay, of the plasmonic resonance, you know if something binds or not on it. But you can have also other info that I will show, because you still preserve the infrared information. And the information that you save is the shape and position of the bands that are of interest for the protein secondary structure. And you can have a really very accurate details on a few molecules on your sample. The other thing that you can do by using these devices is the following. So you can essentially measure in a geometry that is very particular, that is called plasmonic internal reflection. Essentially, your chip is like this. Your proteins are anchored here. You have water. But the point that when your light it comes in this direction, it doesn't penetrate all the samples, but just a few nanometers from the surface. And these few nanometers are basically fully covered by the protein if you have a very well, uniform layer. So the following is that you can obtain information on your protein anchored on the chip exactly with the same resolution that you can obtain for dried samples. So you overcome the water barrier. So this is an example that has been, is a paper, so you can read it. But why I wanted to show you this? Because most of the time we are talking about a recognition, so I'm not a biologist, no? Okay, at all. So, but spike protein and protein, so these are proteins, so the virus circulated, that there is a recognition processes and the internalization of the genome of the virus and so on. So the point is the following. This is a scheme that is the chiave serratura, so the key, I don't know in English how to call key lock. Okay, but this is static if you think about, okay? In reality, these guys in nature, okay, they also vibrate, they have a certain mechanical reaction. And then actually the probability that you have a recognition is not only related, okay, to the shape, but it's also related to the probability that the two things, they lock together. And this is more biomechanical things. Such a kind of forces are forces that can be detected in a very special region of the infrared that is called far infrared, okay? And actually, there is one word package in the PRP CERIC project, this is purely scientific, that wanted to study how is the effect, essentially, of this biomechanics, of these properties, of this special type of vibrations of the key actors involved in the infection, in order to support and to go beyond the concept that there is a recognition, a static recognition, because recognition is always dynamic. Nowadays, there are just simulations on those because nobody studies. So there is a long way to pursue, but I think it's very interesting because could help, for example, to understand how some variants are even more maybe effective than others, which is the effect, for example, of the temperature in the infection, because temperature means a different type of flexibility of the things, and maybe things that we cannot imagine at this stage. So this is one example. For what is concerned, the cytology part, I don't stay longer because it's very technical, but the point is the following. Okay, our technique, similarly to the Raman UV Raman scattering, is more on the side of be ancillary or supportive for those techniques that looks to the infection problem from the phenotyping point of view. Okay, so it's more, I want to it's not a drug-based screening in the classical way, so in the way of macromolecular crystallography or cryo-electron microscopy, but I can study the phenotypes. So this is something that we already started with our colleagues at the HGB, but I just show you this example for letting you know which type of information on the phenotype we can obtain. Okay, first of all, we can measure cells at single cell level. We can measure them both hydrated or dehydrated, it doesn't matter. But each cell has its own chemical profile. Okay, and then if you have two populations, the first thing that you can do, I can discriminate the two populations. Essentially, these points here related to this experiment, these are control stem cells and the differentiated stem cells. This was a study on the glioma, on glioblastoma. But these dots here just summarize in a principal component analysis, essentially plot, this type of cells and this type of cells. So if I can differentiate, but this is not really the very fundamental. Okay, so obviously you can imagine, for example, that I can try to use these techniques for a diagnostic perspective. I will show in this last slide, okay? But there are other information because sometimes, and this is my comment on to the bioinformatics, they just provide the points of differentiation. But the very important question is, what is giving us the differentiation? Because this gives me the bioinformation that I need for understanding, you know, the real difference. In this experiment, for example, when we studied the differentiation of stem cells, there was a very interesting thing. For example, we went back to the spectra, differentiating, and we could observe, for example, that the glycogen level of the other differentiated cell was higher with respect to the stem cell. This is something that nobody noticed before and can provide to people help in understanding, for example, how to prevent, for example, the differentiation to a certain extent. For example, in this case, this would be, the goal for the glioblastoma, but still is an hint for other approaches like the metabolomic one that is mass spectrometry can be much more precise than what I can be, but obviously is also sometimes sensitivity is a good thing, but when you have a lot of things to understand, you need to guide, your information. So it's sort of steps that we can do one after the other. So this is for cellular analysis, and okay, there is just one slide here where I show you some other examples for the study. So we are very sensitive to apoptotic pathways, heat shock response. So there are a lot of studies that you can read. So it's 20 years that I'm doing this kind of things. In the context of PRP CERIC, which is the real challenge. So if I want to use and work with the infected cells and try to understand which is the phenotype, for example, you have an unknown virus at the beginning and you want to understand which is the effect of this virus on cell lines. First thing, I have to take them in a synchrotron. So the first thing is safety. Okay, our goal now with our ICGB colleagues is to try to understand among different inactivation methods, okay, which is the one the less impact on the vibrational profile of the cell, So we tried UV, we tried thermal treatment, we tried, okay, classical paraformaldehyde. At the moment we did these experiments just 1 week ago. The first thing that I told them, forget the thermal treatment. Thermal treatment is always too harsh. So you have for example, in this study here, it is a thermal treatment. When you reach 42 degrees, your proteins are completely misfolded first and then packed in very large aggregates. So you lose all the bioinformation. But it seems that UV treatment seems to be very promising. So first thing is to find a way to inactivate cells without perturbing too much the biological profile, because this will help you to study them still alive, still hydrated at least, in order to get more similar results with other techniques. And then the next step, when we could do this, is to try to test a large varieties of cells treated with different viruses to try to understand which is the phenotyping, which is the profile that a cell should have once infected by a specific virus. This is a phenotyping in such a way that at a certain point, if you have a totally unknown virus that happens and there is an infection all around the world, you can at least, you do some measurements and you at least can identify the major pathways that are affected by this virus. This is one idea, and you can do obviously in combination with many other techniques. So this is the first approach. So we do the same things with the tissues, okay? So you can work at cell level or you can work at tissue level. I would say that PRP CERIC is mostly for cytology more than histology, but still we had the opportunity, for example, during the coronavirus to measure some samples from the lung for patients that have died with the corona. Obviously, they were fixed in paraformaldehyde and so completely inactivated. But there are also possibilities to test the effects of the virus in some organs or districts. So this would be useful to use like, an histological approach on large tissues. For the nano stuff, it's the examples that I showed you before are mostly related to microscopy or spectroscopy. On nanoscopy, it's very demanding up to now to work with biology because the technique was invented by physicists, and the physicists, the maximum of complexity that they can understand is a silicon piece with some circuits on top of it. But biology is much more complex, than this. So we did some experiments, and just I want to show you this example. This is a study that we started with in collaboration with the joint GRC. Okay, this is an European center in Lombardia. Okay, And the idea is the following. Most of the time, your vaccines they are administered with adjuvants. Okay, like aluminum adjuvants is one of the most popular. The point is the following. I have my molecule. My molecule works perfectly as a vaccine in suspension, for example. And then I put it with these adjuvants for promoting the immune response. But the point is, which is the folding of this guy on the adjuvant? Because sometimes you don't have a direct correlation between, most of the time you don't have, between the activity of this molecule in suspension and when it is co-administered with an adjuvant. So you expect an enhancement of the effect, but you don't have it in reality. The point is that each guy that interacts with the surface, it changes because you have surface forces, okay? So, and the study was based on the understanding of this, on the folding state of the receptor binding domain of SARS-CoV-2 on aluminum adjuvants. And then we could appreciate on these adjuvants, so at the nanoscale, that there was effectively a misfolding of the protein on those formulations that were less performant with respect to the others. We didn't publish this work yet, but we are obviously in tight contact with several pharmaceutical companies because this will be a challenge with respect to the present knowledge. Obviously, we do not have the sensitivity of X-ray diffraction or TEM because I cannot tell you exactly which part is misfolding because my information is general, but is an information that I can achieve essentially in the real condition and on the real pieces. You cannot do the same, neither with TEM or NMR or X-ray microscopy, X-ray crystallography. So the other example I don't tell you. This is more on the other things. I want just to tell you something about for reconnecting with the point of care analysis. Okay. So one of the parts of the project, of the PRP CERIC project that was on the work package that also it's mostly led by the omic part, is the attempt to exploit infrared spectroscopy for doing some easy device for point of care analysis. So this is a work that has been done in collaboration with Halifax. So essentially Halifax has all the patents for this because we sell our knowledge to them. So you know how you work with companies is like this. But essentially, we developed with them a system that is on the market now, on the hospitals, that is for bacterial strain recognition, essentially. You know that nowadays, these things are done most of the time with the mass spectrometer. Use mass spectroscopy. Very sensitive, but it has a very big drawback. It's very expensive. It's very expensive. So a very good mass spectrometer can cost like 500 kilo euros. Maybe for this application, 300. 300. So this guy here, it costs 20 kilo euros. You can use everywhere. You do not need to have a environment control like most of the time in the time of flight. Mass spectrometer you need is fundamental, otherwise your stats doesn't have any sense. So the idea is to try to exploit the same kind of technology in order to provide easy and fast tools for point of care analysis, also for the viral infections. So
you use this mass spec as gold standard?
Yes, but in general, this is bioinformatics. So essentially, you build your database, you start with sprays that are known, and then you build your space of variables. And then when you have the unknown, you put in these clouds, colored clouds. That means a specific guide.
Yeah, it's funny because the market of guys use us to validate the data.
Possibly.
So it's just a chain.
It's a chain. No, but it's okay. You need to validate. Yes, I saw. Okay, in one of the presentations for the selection of the bioinformatics guys, there was someone that showed this kind of thing. But it's normal. This is nothing secret. This is the way the different techniques can talk each other and then support each other. So, what we will do with the project PRP CERIC, we will further strengthen our capabilities in the nano IR in the imaging part. So we will acquire new instruments. But maybe could be interesting for you, we acquire one guy that is nowadays not on the market, it's only one in Italy, that have a lateral resolution between microns and few nanometers, like 500 nanometers. That is this range of lateral resolution that is closer to fluorescence microscopy, for example, that is again a sort of phenotyping approach that is mostly used to the HGB guys. Okay, so essentially we will have all the spectral resolution, the lateral resolution for characterizing the sample. So this is my group, very large. So thanks for your attention, and I know that it's very complex, but as for me, your talks.